form back into the bilayer. It's an excellent structure. I mean, excellent structure. So this structure allows for proteins. Wait a minute. Up to this point, what have we learned about proteins? All right. Yay! Let's go back to our general bio. Let's regress for a few minutes, okay? And let's think back to a cell with a nucleus with the DNA. Okay? All of our chromosomes. Okay? Does that kind of look familiar? I'm not an artist, y'all. Okay. Now, on my DNA are my genes, not the ones we wear. Okay? But how many, we talked about this the other day, how many molecules of DNA do I have in the nucleus of this cell? 46. What we know of as 23 pairs. Is everybody with me so far? This makes for a huge amount of genetic information to be held. Does every cell of your body, does every cell have to make every protein coded for by every gene on your DNA? No, it can't. All right? It's not possible. Your genetic code that you have only does about 30% of what you actually carry. All right? And from person to person, we differ very little. Because we all have to make the same proteins. I have to make insulin. You have to make insulin. Okay? We have to make the same proteins. So far, what we know is that proteins can be a product of a cell. So if I have a gene that's going to code for a protein that this cell is supposed to make, it will come out, make the protein, I'll get the protein, and it'll move to the cell's membrane, and then it'll go out into my extracellular fluid and be used by the body. On my DNA, I'm going to also have genes that will code for proteins that I need inside my cell. What kind of protein would I need inside my cell? Ever heard of a lysosome? Okay, I'm going to have a gene that will code for something that stays inside my cell, such as a lysosome. Now, I'm going to have a gene that's going to code for proteins that need to be in my plasma membrane. So now I'm going to code for the proteins that will exist in my plasma membrane. Is this making sense? Because of all of this genetic code, the only thing that it can do is make a protein. So, for the proteins that are going to become important in that cell membrane, we're going to find that they get embedded in my plasma membrane. We're going to have proteins that can be integral or peripheral, either intrinsic or extrinsic. If something is intrinsic, you do it for what you get from it, okay? It's all about being on your inside, all right? But if you do something, say for, I don't know, some charity or whatever, that could be extrinsic. It's outside yourself, all right? So what they're trying to tell us is that we are going to have proteins that can be intrinsic, going deep into that membrane, and they usually poke out of the bottom here. Because if I have those, it can help give structure, such as what you see right here, holding on to the cytoskeleton of the cell. This would be a protein attached. It could be attached to this cytoskeleton, and it would be holding it in place. That's integral. That's intrinsic. That's its only job. It doesn't do anything else. That's it. Because proteins are specific. Okay? So if that protein was made to attach to the cytoskeleton, that's what it wants to go do. It wants to help be a structural protein. If it is extrinsic means that we could have those proteins, it could be actually poking out on the outside of the plasma membrane. Function will be dependent on the shape. It could be a protein, and that membrane that would have the glycose, these sugar chains, they could be used for communication, receptors, stuff like that. So this is one way, all right? So if we think about the types of proteins, okay? We have those that are going to be labeled integral and peripheral. All right? Most of the time, their job is membrane structure. The proteins, when we mentioned just a few minutes ago, the glycoproteins, the glycolipids, these are for the cell communication and the cell receptors. Reasons that we need these, it can allow the cell itself to communicate within itself. It can allow the cell to talk to another cell. And one of the things that'll be important, These are going to function in receiving of stuff like antibodies, which are going to be important in the immune system. They can function as receptors for hormones, which are going to pretty much run the entire body, okay? But their job is usually communicating and receiving. Now, another type of protein, okay? So another type of protein are the ones that are the attachment. All right? We got the glycose. Okay? So now, if we look at the attachment proteins, one of the things that I keep saying, cells are going to make up tissues. Tissues are going to make up organs. Organs are going to make up organ systems. Organ systems make up the organism. This means that our cells have to attach either to each other or in some way to another cell. Maybe the one that's making up their connective tissue wrapping. We are not in any way disorganized. As humans, we are very organized beings. We can have cells, proteins. Notice how they're being represented as being within that plasma membrane, and it can attach to something inside the cell. It can attach to something outside the cell. They're very specific. What do you notice being represented here? Their shape is specific for something that fits only into it. Proteins will always be very specific. They'll have their one function, and that's pretty much it. So we've got attachment proteins in that plasma membrane. Now, transport proteins. This is the concept that is the most important. All right? Transport proteins. We have to transport something across the cell's membrane. In the transport proteins, we are going to have proteins that function as a channel, proteins that function as a carrier, and proteins that will be an ATP powered pump. When we talk about the channel proteins, now this is where it gets exciting. Note my terminology. Non-gated ion channels. So this tells me that I'm going to have a protein embedded into the cell membrane, and it's going to have a channel that exists in that protein,